Hi, and thank you for joining us for Chardan's Virtual Ophthalmology Summit. The focus of this installment is on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to introduce Paul Bresge, CEO of Ray Therapeutics, and Peter Francis, Chief Medical and Chief Scientific Officer. Paul, Peter, thank you very much for joining us.
Thank you very much for the invitation, Daniil.
Of course. So the format for this session that will follow is going to be a fireside chat. So I'll present a number of questions, and then we'll have a discussion about your company and the programs. So with that, let's go ahead and get started. So if you both could take a minute to introduce yourself, and Paul, maybe if you could spend an extra couple more minutes to introduce Ray Therapeutics to the audience.
Sure, of course. Thank you so much. So yeah, Paul Bresge, I'm one of the founders and the CEO of Ray Therapeutics. Very pleased to be attending the conference. Thank you for the invitation. I'll give you some background on the company. We are very much a California-based company. We have offices and labs in Berkeley, where I am right now, and then we have labs in South San Francisco. And then we have a central testing facility, which we call the Vision Research and Assessment Institute, and that's located in Southern California. The mission of our company is to transform the lives of patients with retinal degenerations by restoring vision using optogenetic gene therapies. We have two lead assets. One of them is already in clinic, and that's RTX-015. And it's in clinic for retinitis pigmentosa and other inherited retinal diseases. The second one that we are in already is choroideremia. And then we have another follow-on program that will be in clinic towards the end of the year, and that's RTX-021 for Stargardt's and the advanced form of macular degeneration, geographic atrophy. We're the first bioengineered optogenetic medicine that has been optimized for human vision. The protein that we're using is exquisitely light sensitive, so patients do not have the need to wear additional light-enhancing eyewear. And we are looking to restore the higher visual functions, such as visual acuity, contrast sensitivity, visual fields, and mobility. We've been very fortunate to be funded approximately $122 million. The company was incorporated in early 2021. We did a Series seed round of $6 million that was led by 4Bio Capital. And then we did a Series A in 2023. It was a $100 million Series A led by Novo. And in the syndicate, we have Deerfield, Norwest, Platinus, MRLV, and 4Bio Capital. We've also been very grateful to have been awarded about $16 million from the California Institute of Regenerative Medicine to support both the 015 and the 021 programs. Because of the funding that we've received, we feel like we have really had the opportunity to design and build the company for success. Peter will talk about the Vision Research and Assessment Institute, but essentially it's allowed us to really standardize and develop endpoints. It's given us great access to enrollment in our clinical trials. We've had excellent interactions with the agency. We have a very defined pathway to approval. And again, Peter will talk about that later on. And then in terms of CMC, which is so important in gene therapy, we are very much ahead of the game here. Before we even dosed our first patient, we had already established our commercial process and our commercial scale. So we're, as I said, in great shape with manufacturing. And in fact, we're already preparing for commercial manufacturing for our RTX-015 program. In terms of the pipeline, as mentioned, retinitis pigmentosa and choroideremia are our first two indications. Stargardt's, we intend to be in at the end of the year, and then moving towards geographic atrophy in around Q3 2026. What's very important to note about our therapy is that it is completely independent of the underlying genetic mutation. So we're able to treat very large patient populations, even within these more rare diseases. So for example, in retinitis pigmentosa, the prevalent population just in the United States alone is approximately 50,000 patients. And when we look at the other inherited retinal diseases as a bucket, that's approximately another 50,000 patients. For Stargardt's disease, the prevalent population is approximately 20,000 with a very high incidence rate. And then, of course, we all know that in geographic atrophy, that's a huge population with approximately 1.5 million patients in the United States alone.
Mhm. Got it. Excellent. Thank you, Paul.
Of
And
course.
Peter, if you could spend a minute or so to introduce yourself.
Peter Francis, I'm the Chief Medical and Scientific Officer at Ray Therapeutics. My background is as an MD PhD ophthalmologist and retina specialist, and I've been working in the biotech industry for approximately 2 decades, principally in gene therapy and ophthalmic disorders. Thank you for the invitation.
Of course, excellent. All right, so let's jump in and talk about your program. So the lead two lead indications, RP and choroideremia. What can you tell us about those diseases? What is different and similar between them, and how are you able to utilize the same product for two separate indications?
So they are in many ways quite similar disorders. So both are incurable, progressive retinal degenerative diseases where patients unfortunately lose vision because the light sensing cells, the photoreceptors, are progressively lost. The underlying mechanisms of retinitis pigmentosa and choroideremia might be slightly different, but they converge on the fact that ultimately the photoreceptors are lost. And these light sensing cells, photoreceptors, don't have the capability in the adult to regenerate. They are responsible for light sensing and sending a signal to the brain. So when the photoreceptors are lost, blindness ensues in that region. Our treatment looks to restore vision using optogenetics in the surviving cells of the retina. So in the retina, in addition to the photoreceptors, which are lost in these conditions, there are a number of other cell types, for example, retinal ganglion cells and bipolar cells. And these cells survive the disease process very much into the latest stages of the disease. And so optogenetics can make those surviving cells light sensitive. And then light coming from the environment into the eye, focused on the retina as it normally is through the lens system of the eye, actually then makes those cells respond to the light, and they send a signal to the brain. So you can see that although the conditions of RP and choroideremia are distinct in the sense that they're caused by different mutations in different genes, they are unified by this loss of photoreceptors, and yet these other cells remain, and our optogenetic approach can make them light sensing. And so our treatment should work in just as well, effectively, in an RP patient as well as choroideremia. And for that matter, many of the other inherited retinal degenerations, which are rarer than those two, but nonetheless, the treatment should also work. And so in time to come, we may well incorporate those patients in our clinical evaluations.
Got it. Okay, excellent. So let's take a step back for a second, and if you could explain what specifically an optogenetic approach is in broader terms, and why is it necessary for these particular indications, maybe with an emphasis on the target population, and specifically about your product RTX-015, what makes it unique that compared to other optogenetic approaches?
Okay, well, perhaps I'll talk about the overall optogenetic approach, and then I'll hand it to Paul, and you can perhaps talk about our differentiation. So optogenetics refers to a field of research which is now almost 25 years old. Nature has evolved channel rhodopsins, which are an optogenetic protein. These are typical transmembrane channel proteins that are found in all species, and they allow the ions to cross the cell membrane. Channel rhodopsins are unique in nature because the channel through which the ions can flux is gated, and that gate is opened by photons of light. So these proteins can be expressed in cells. They make their way to the cell membrane, and then photons of light that are incident on these proteins open the channel, and you get a flux of ions. So neuroscientists principally have taken these proteins, expressed them in neurons. You can shine light on these neurons expressing the channel rhodopsins, and you can generate what's called an action potential, an electrical signal. And so they've been used diagnostically and very, very well to map brain circuits and so on. So what we've done is we've taken that technology, that very same technology, and we're now expressing them in the retina of the eye, as I described, in neurons, in cells that are not normally light sensing, and we can make them light sensitive, as I described in the earlier answer. Light coming into the eye, focused on the retina, then excites these channels. They allow flux of ions like calcium. They generate a signal thereby, which is then sent to the brain for interpretation as vision. So that's the general field. So it's a gene therapy in this instance that's delivered to the eye. The gene therapy results in transduction of the cells, which then express this particular protein forever. We think pretty much forever, or for the lifetime of the patient, and then renders those cells light sensitive so they can get formed vision. So I'll hand on to Paul now, maybe to talk about our key differentiation.
Sure, of course, thank you. So yeah, just really as I mentioned before, we are the first bioengineered optogenetic gene therapy. So the previous generations have essentially relied on the wild type proteins. Those proteins do provide light to the remaining cells, and that's how we have such great proof of concept in the field. But ours being an engineered protein specifically for human vision restoration should provide the potential for those higher visual functions that I mentioned earlier. We're also using the most light-sensitive protein that's found in nature, so that eliminates the need for the light-enhancing goggles that some of the other programs have had to incorporate into their clinical development. Also, we're addressing patients that have a baseline of potentially as good as 2,200, so that's on the higher end of patients with advanced disease, so that potentially allows us to address a much larger patient population than those that are addressing patients at the very, very severe end of disease. Peter, anything to add to those points?
I think that's excellent.
Okay, great, thank you.
Got it. Excellent. All right. So in terms of the construct, the RTX-015, it utilizes AAV7M8 vector that's licensed from Adverum. Can you talk about how that partnership came about and what made you choose that vector?
Sure. Peter, you want me to start?
Please go ahead. Yes.
Sure. Well, I mean, Peter, has certainly a great deal of expertise, in the field, given his background. We were very confident, from the beginning that 7M8 was going to be the appropriate vector of choice for us, and it has turned out to be, an excellent choice, and we've had a great partnership with Adverum. So obviously, we compared 7M8 to several other available vectors and went with 7m8. Peter, anything to add to that?
Yeah, so our approach, the approach that we wanted to take to delivery of our agent was the intravitreal route, which is a simple office-based procedure by which you can deliver biological therapeutics to the eye, because it allows full distribution of the therapeutic to the whole retina, rather than a subretinal approach, which is much more of a localized and involved surgery. So with that in mind, we were very conscious of the fact that the normal AAVs, AAV2, AAV8, and so on, typically don't transduce the retina following an intravitreal injection very efficiently, if at all. But we were, as Paul said, very much aware of the development of these newer vector variants from directed evolution like 7m8 that can get enhanced transduction of the retina following intravitreal route. And I have to say that our preclinical studies have been impressive in terms of the biodistribution and expression of 7m8 vectors, and the safety profile at the doses we're using looks very good as well.
Got it. So what one other potential advantage that you may have is the clinical experience, right, that Adverum had with 7m8. They took it through a number of patients already. They spent a lot of effort to come up with the most appropriate steroid regimen, prophylactic steroid regimen, that they're taking to the pivotal trials. What are the key learnings, or are you looking at their programs and their findings in terms of safety and so on that you are incorporating into your programs, into your clinical trials?
So I think we have a very good relationship and a very good collaboration with our colleagues at Adverum, and working with them, and also world leaders in intravitreal AAV gene therapy from the clinical perspective. We were able to develop a robust prophylactic steroid regimen that's very closely aligned with that which we understand Adverum is utilizing. So we really are pleased with this. The tolerability of the regimen has been excellent in our patients, and it combines an oral as well as a topical steroid regimen that we continue for a number of months and is slowly tapered off. And in our patient population, who are generally healthy patients, otherwise, other than their eyes, and slightly earlier generation or earlier age of patients than perhaps the macular degeneration patient population, we've been very pleased with how our regimen has performed and the acceptability of that.
Okay, yeah, makes sense. All right, and as you guys alluded to, for optogenetics, this is obviously not a first-gen approach. There's been a proof of concept established in patients, in humans, so that's a great start. But specifically for RTX-015, can you maybe highlight some of the preclinical data, and what models specifically you used that gave you confidence in that particular program's potential success?
Absolutely, yes. I think it was central to the formation of the company, and it's been central to our success. The absolutely comprehensive nature of our preclinical pharmacology. So our inventor and then reproduced in studies that we've overseen. We've seen demonstrable and repeatable, reproducible evidence of creation of visual acuity and contrast sensitivity, navigation, and peripheral vision in animals that were congenitally blind. So they were born with no vision whatever. That's why I use the word creation. It's not in this instance vision restoration. It's actually creating vision in these. And we've looked, we and others, and in particular our inventor, have looked at the model systems in great detail. Firstly, when we think about how you measure vision in an animal, there's been a lot of work done using electrophysiological measures, which are don't really tally with or don't really translate into true vision. There have been a number of behavioral tests that can be done in animals, but actually most of them are not particularly validated or reproducible. Whereas the work we've done has really been based around a very objective measure called the optomotor response, which is an innate measure in the animal. It doesn't learn how to perform the test. It's something that the animals will do. It can be measured by independent observers, in some instances actually using head tracking software and hardware. So no interpretation needed. So we get very robust, reproducible, and sensitive measures and quantification of the level of vision, as opposed to just an on-off response. So endpoints wise, we're pretty convinced that we have something which is very robust and reproducible. In terms of the animal model, our optogenetic approach has been tested in several different animal models. Commonly, RD1 and RD10 degeneration animal mouse models have been used, and we certainly see a qualitative outcome in those models using our optogenetic approach. However, they fall down because you can't really quantify the response, because these animals degenerate in a very highly variable fashion, and measuring responses from them can be very challenging. So we've used what I think has now become the gold standard preclinical model, Paul, which is the triple knockout mouse, which is this congenitally blind mouse where we can express in the cell type of interest, and we can reproducibly over time measure these optomotor responses, giving us a really very nice data set that we've expanded beyond just a test versus treated versus untreated to now a full dose ranging analysis across all therapeutic doses. And we've also been able to look out for durability. So we've been able to look out to 12 months or so in this animal model. In terms of further translation, we've been able to look at biodistribution and expression in the primate, the non-human primate in this instance, which has also given us an excellent insight into toxicity and so on, given that the non-human primate eye approximates to the human better than any other species. So
Right.
yeah, I think we have a robust, very robust pharmacology and toxicology package.
Got it. Excellent. Thanks. That's a great overview. And one more question on preclinical. In terms of kinetics, how soon do you observe effect from the therapy, and how would you expect that to translate into humans?
Yeah, we see benefits in terms of vision improvement as early as a week or two following treatment, but they increase over time. So we typically are picking up, 3 to 6 months. We start to see the maximal benefit in the animals. We think that because the kinetics of gene expression are similar, that we can expect, a relatively early report of visual improvement to probably a minor degree in treated patients, which then over the ensuing months, 3 and 6 months, would continue to improve. The other area that we think you may get increased efficacy over time probably stems from cortical adaptation. So the brain learning essentially to see more and more from the visual stimulus it's now receiving from the eye. And we've seen this in other studies like the Spark Lux Turner studies, where up to 3 years afterwards, patients continue to see some degree of visual improvement, probably driven by cortically and cortically driven processes improving the interpretation of that visual signal. So that gives you a nice time course. It's sort of very much an early efficacy readout, which contrasts with many of the rare ophthalmic disease treatment studies where you may have to wait 1 to 2 years before you can really detect a significant difference.
Got it. Okay. And your 015 program is in the clinic now, right? You're dosing patients in the phase 1/2 trial. Just to understand that trial, if you can walk us through the design, how many patients, and do I understand correctly that this trial includes both the RP and choroideremia patients?
Yes. Paul, do you want to take this one?
I think that maybe let's take a step back, because what's so key to the way that we have approached clinical development is how we're testing these patients. So we have to recognize that we're testing patients who are 2200 or worse, and while we're using some of the best clinical sites in the country, we recognize that patients who are so advanced in disease, it takes a great deal of time and particular expertise in order to test these patients with the different endpoints. So we built out this Vision Research and Assessment Institute. It's in Southern California. It's very close to the John Wayne Airport, which is a very low stress airport because for patients with low vision, just traveling through an airport can create an enormous amount of stress in and of itself. And we've really staffed this center with some of the country's top low vision experts to help us define appropriate endpoints, to refine, these endpoints, to standardize them. So we're doing, some of the better known endpoints, like, visual acuity and mobility and contrast sensitivity. And then we're also exploring some, very unique endpoints as well. And that has been very, very well received from the FDA. They've really applauded us on the work that we've done. We also did extensive observational run-in studies where we had patients come in for testing and retesting so that we could get the noise levels down. I think one of the things that has really plagued programs in retina has been the great amount of variability in these patients. We're looking at very heterogeneous diseases, and patients present very differently. And what we've been able to do with this Vision Research Institute, through the observational studies in particular, is really reduce that noise level. And again, we've aligned with the agency on what these endpoints are, and also what would be clinically meaningful. So that's I think very important to understand how we've approached the actual clinical program. In terms of the design, Peter, do you want to talk about how we designed the typical 3x3, and then what our thinking is moving forward in RP, and then we can move on to choroideremia?
Yes, and we're very conscious of in the phase 1 study the need obviously to assess patients who have advanced retinitis pigmentosa. So we're very careful to include therapeutic doses throughout. As Paul mentioned, this is a modified 3+3 design, by which we mean it's serial enrollment of a group of 3 patients, with initially a sentinel patient and then 2 others. Then we review the data with our DSMC and decide to dose escalate and proceed in that fashion. It is uncontrolled. It is open label, as would be typically expected, and it's a single eye treatment study. Nonetheless, we're able to deploy not only those safety measures, but also all of the efficacy measures that we're discussing today, with a reasonable expectation that we should see something in our patients. Now, by their nature, many of them are pretty advanced, and so I think that would naturally expect to limit the benefit. But we've enrolled a spectrum of patients within our enrollment criteria. We're enrolling patients with retinitis pigmentosa, 2200 or worse vision, down to around about 28,000. So just for those who may not realize, 2200 is defined as legal blindness in the United States. So it's basically legally blind patients with RP, but not those patients who have no light perception, which is rare, but they probably represent a slightly different group and much less likely to respond. So enrollment has been very brisk. We're very grateful to firstly the patients and their families, and obviously our investigators. Both groups are incredibly motivated, and so we've had some excellent enrollment, and now we're, as Paul mentioned, we're still enrollment has been brisk enough that we're still collecting data. Many of our patients haven't even completed 6 months following treatment, so we'll be certainly talking about efficacy in short order. But right now we're really just collecting sort of the efficacy data and safety data and very early efficacy readouts.
Okay, got it. Makes sense. And just to clarify, Peter, is it a single trial that is enrolling both RP and choroideremia patients, or are there 2 separate trials?
No, it's the same trial. The FDA actually gave us a choice. We said to them, hey, would you like us to submit a separate protocol? And they said, it's entirely up to you. We'd be happy to have this as part of the same trial. So we enrolled the low dose and the mid dose patients in the RP study, and then we started enrolling choroideremia patients at that mid dose once we had enough safety information to give us confidence that that was a useful dose to go to. I think I alluded to the fact that we've had excellent interest for our RP patients. I have to say the Choroideremia Research Foundation, we have really good, strong connections with that group. They do excellent patient advocacy work, and we recently attended their national conference a few weeks ago, and I have to say there was really considerable interest from the choroideremia population to actually get into our study.
Excellent, got it. And in terms of when you're getting closer to data readouts, what efficacy signals would you consider to be a home run for your company and for patients?
I think the home run is an easy one. The strength of the retina field is that we have very good, validated, fully approvable endpoints in best corrected visual acuity and multiluminance mobility testing, and we understand what is the clinically meaningful level of improvement, which in the case of visual acuity is greater than 0.3 logMAR improvement, and in the case of mobility testing is 2 or more light level improvement in sensitivity. So any of those would be a home run. I think, however, it is, as always, a lot more comprehensive and complex than that. And so we would also expect to see improvements in many of the other visual testing that we do, which correlates with visual acuity and multiluminance mobility, by which I mean in terms of central visual function tests. In terms of acuity, you have contrast sensitivity, for example, and then in terms of mobility, you would expect to also see changes in visual field sensitivity and full field threshold testing. So it would be that total package of data that's very supportive and compelling biologically in this uncontrolled phase 1 study that would be the home run.
Mm-hmm. Got it. Makes sense. And Paul, I have a quick follow-up question on that centralized facility that you've built in Southern California. Can clearly see the benefit to your company, but at the same time, there are a number of other small biotechs who are looking at other rare inheritable diseases. Do you get any interest from others to work together at this facility? That's something that you could potentially monetize or loan to others to use and make the entire field of IRD more standardized?
Yeah, it's a great and very appropriate question, because we are very excited with the number of companies that are doing retina studies that have approached us, recognizing that what we have done, is really unique and really necessary. So it's come in two forms. It's come in, one form, competitors telling us, very directly, thank you so much for paving the way. We're going to copy exactly what you're doing, and we're going to build out our own, central testing facilities, which we see as fantastic, because again, it's very important for patients and for the, potential success of these retina studies that there are, drugs approved. I was at a conference recently, and I heard, somebody say something that has really resonated, and that is, sometimes the trials fail the drugs as opposed to, the drugs failing the trial. So the fact that we have, I think, prospectively set this up for success and that others are following, I think, is a very good thing for the community. So that's been, one, sort of one approach, and, we heard it very directly. And then, whenever we talk about this Vision Research Institute at conferences, which Peter and I, attend and present at regularly, we are approached, all the time by companies asking us if they could have access. And that's not only biotech companies, these are also CROs that have asked us if they could, have access to the facility for their, for some of the studies that they're working on. And, it's something that we're considering, not so much, at this point, Daniil, in terms of, we're not thinking about it necessarily in terms of, how do we monetize it, but we're thinking more about how do we, support the community, and how do we support endpoint development, through the Vision Research Institute. And, I think that, we're playing a very important role here. And, there's a strong possibility, if it's helpful, to those other programs and to ours to bring, additional patients through, sometimes with different diseases, the more learnings that we have, and the more that we can share, the better. But of course, we remain very focused on, our clinical programs. As mentioned, we already are in 2 indications. We'll be in a 3rd one, just this year. So we have to keep the team there very, very focused on the work that they're doing. We've also built it out in a way that it could be replicated very easily in, additional, places in the United States, or, we're thinking of a global study in Europe as well. And it's also, why we have, developed, SOPs around all of the testing, so that we can have successful tech transfer and, potentially build out additional sites.
Mm-hmm. Okay, got it. Makes sense. Last question on 015 program. In terms of the next steps and the potential timelines to initial data readouts, can you share that?
Yeah, I mean, it's still, as Peter mentioned, early days for us. We've had our low-dose patients, come through for their 3 and 6-month visits. We've had some of the mid-dose patients come through for their 3 and a couple 6-month visits, and we're starting to have the higher dose patients come through. But as Peter mentioned, we're excited about the data maturing, and being a private company and not really having the need to raise funds. We have the luxury of being quiet, and being very focused on on the trials, and very operational. But of course, we are thinking about at what point, and at which conference would it be best for us to present this data. But as I mentioned, it does it has to mature. We also have to make sure that we're demonstrating not only safety and efficacy, but also durability. So we're obviously factoring all of those different components into the equation of when we announce what the data is from the study.
Okay, got it. Makes sense. Let's spend a few minutes talk about your other program, 021. So that's in development for Stargardt and geographic atrophy, obviously bigger indications, especially GA. But in terms of the construct itself, what is different between 015 and 021?
Yeah, maybe Peter, I'll let you talk about it, but maybe just one thing to mention at the front. This was very, very much driven by the science. So, we're very excited, with the potential of targeting, the bipolar cells. And that's what 021 does. That's essentially the differentiation between 015, which targets the retinal ganglion cells, and 021, which targets the bipolar cells. The difference in the construct, is essentially that 021 has a on bipolar specific promoter. That's really the only difference. So it does target those bipolar cells. Peter, do you want to talk, a little bit more about the 021 program and maybe even the difference that we saw in the preclinical results between 015 and 021?
Yes, I mean, there's a theoretical advantage to being able to go after the on bipolar cell in the retina. So the reasons being that there are more of them, so potential for higher resolution, and they're also a little bit more upstream in terms of retinal processing of the image, and so they could give a theoretical advantage there as well. The problem is that in many conditions that we've talked about today, RP, choroideremia, and many of the inherited retinal degenerations, particularly in this target population of patients that would benefit from optogenetics, the more advanced cases, most of the bipolar cells are lost, and many of the on bipolar cells get remodeled as off bipolar cells. And so it becomes very difficult to really utilize them as the cell type of interest. But it so happens that in Stargardt's and geographic atrophy, it does appear that these bipolar cells do survive into this in this disease in this patient group, and so we can legitimately try to use optogenetics with bipolar cells. Then come the technical challenges. As Paul mentioned, we would need to go after or need to treat the on bipolar cells. And so you definitely do not want to treat on and off, because the off bipolar cells do what they sound like they do. They actually switch the signal off. They're involved in center surround phenomena and so on. So you need to be able to specify on bipolar cell expression of your protein, and that hasn't really been possible until recently. And we in-licensed a proprietary novel cell-specific promoter. So we can use the vector, the AAV vector, to transduce the retina, and then we can say we only want to express it on bipolar cells via this particular promoter. It is a human-optimized promoter that is also highly expressing, and that's really important. So early attempts to generate on bipolar cell promoters generated promoters that were leaky. They didn't always just express on bipolar cells, and they were very weakly expressing. And so we seem to have conquered that, and our preclinical studies back that up. As Paul mentioned, we've really, by comparison with 015, we see 021 outperforming, which is what you would hope. So more light sensitivity and higher resolution vision, more contrast sensitivity in the animal testing. So we're very enthusiastic about how this is going to look in humans. In particular, contrast sensitivity loss is a real feature of geographic atrophy, and would be of an enormous material benefit to patients in terms of the quality of their vision, to be able to restore that to a significant degree. And our data really supports contrast sensitivity improvement as being a potential here. So we're very enthusiastic. And of course, the read-through from the 015 program is significant. We're using the same optogenetic protein, and given what we've seen in the phase 1 study, we'd really like to get into the 021 program as soon as possible.
Okay. And when you say as soon as possible, are you expecting this to be in the clinic sometime later this year, or are you?
Yeah, correct.
Okay, excellent. All right, just a couple more housekeeping questions. So Paul, you mentioned the raises to date, 100 million Series A in 23. Also a significant CERM grant. With all that funding, what milestones do you expect your current cash to carry you through?
Actually, very significant milestones in terms of data readouts as well as manufacturing. So we are very grateful, particularly in this funding environment, to have such extensive runway and to be able to really meet very meaningful milestones, essentially for all of the programs. So yes, and again, I have to say how much we appreciate the investment from the different Series A and seed investors, as well as California Institute for Regenerative Medicine. In these times, in particular, to get non-dilutive capital, is very very exciting and important, and also very validating for our approach. On this last grant where we were awarded $8 million, it was the score that we received was a unanimous from all reviewers, pretty much not pretty much, but on all sections. So we got really essentially a perfect score, which of course was very validating from a program perspective.
Excellent. Yeah. Well, great. And guess the last question, as you are taking one program through the clinic, getting ready to enter the clinic with another, what do you think investors might still be overlooking about Ray Therapeutics?
I'm going to use the word grateful, but I'm grateful that I don't think that investors are missing anything. It's been quite remarkable how much inbound interest there has been from investors. As mentioned, we're not actively raising money, and yet we are talking to investors, and in large part it's from people such as Chardan and other groups that are introducing us. And I think also just sort of buzz amongst the biotech community and investors that we have a very a potentially very, very promising program for patients with very high unmet need.
Yeah. So, well, I think this brings us to the end of the session. Again, Paul, Peter, really appreciate the time to join us, and thank you for a great discussion.
Thank you very much. Great to see you.